Good morning. I am Madhurima Datta, Zoology and Biology Educational Content Creator. Now, content for B.SC Zoology Honors, M.SC Zoology, CUET PG and CSI Net Life Sciences. Paper Non-Chordates or Invertebrate Zoology or Diversity of Life Form and Unit Platyhelminthes. And the topic is General Characteristics of the liver fluke and liver fluke life cycle in human. At first, we know about the class Turbellaria. Turbellarians are most free-living worms that range in length from 5 mm or less to 50 cm. They can be found under object in marine, freshwater, and terrestrial habitats. Turbellarians are often distinguished on the basis of the form of the gut and pharynx. Polyclads have a folded pharynx and a gut with many branches. Polyclads includes many marine forms of moderate to large size and a highly branched intestine in correlated with larger size in turbellarians. Turbellarians typically are creeping forms that combine muscular with ciliary movement to achieve locomotion. Very small planaria swim by means of their cilia. Beating of the epidermal cilia in the slime tract moves the animal forward, while rhythmical muscular waves can be seen passing backward from the head. Large polyclads and terrestrial turbellarians crawl by muscular undulation much in the manner of a snail. Next class, Trematoda. Trematodes are all parasitic fluids, and as adult they are most all found as endoparasites of the vertebrates. They are chiefly leaf-like in form with one or more suckers but lack the opisthopter present in the monogeneal fluids. Next, other structural adaptation for the parasitism are apparent. Various penetration gland or glands to product cyst material, organs for adhesion such as suckers and hooks, and increased reproductive capacity. Otherwise, trematodes share several characteristics with ectolecical turbellarians such as a well-developed alimentary canal. Of the subclass of Trematoda and subclass Aspidogastria is the least well-known. Most parasites in this group have only a single host, usually a mollusk. Next, liver fluke in humans. Introduction. Clonorix. Clon means branches and orchis means testis. Is the most important liver fluke of human and is common in many regions of Eastern Asia, especially in China, Southeast Asia, and Japan. Cats, dogs, and pigs are also often infected. Next, structure of the liver fluke. The worm vary from 10 to 20 millimeter in length. Their structure is typically of many trematodes in most respects. They have an oral suckers and a ventral sucker. The digestive system consists of a pharynx, a muscular esophagus, and two long unbranched intestinal ceca. The excretory system consists of two protonephridial tubules with branches lined with phlegm cells. As in common in fluke, about 80% of the body is devoted to reproduction. The reproductive system is hermaphroditic and complex. They have two branched testes that unite to form a single vas deferens, which widen into a seminal vesicle. The seminal vesicle leads into ejaculatory duct, which terminates at the genital opening. The female system contains a branched ovary with a short oviduct, which is joined by ducts from the seminal receptacle and vital area at an ootype. The ootype is surrounded by a glandular mass that is Mealy's gland of uncertain function. From Mealy's gland, the much convoluted uterus runs to the genital pore. Cross-fertilization between individuals is usual and sperm are stored in the seminal receptacle. Next, life cycle of the liver fluke. The normal habitat of the adults is in the bile passageway of humans and other fish-eating mammals. Eggs, each containing a complete miracidium, are shed into water with the feces but do not hatch until they are ingested by the snail Paraphosphorus or related genera. In a snail, a miracidium enters the tissue and transforms into a sporocyst, which produces one generation of radia. A radii is elongated with an alimentary canal, a nervous system, and excretory system, and many germ cells in the process of development. Radii pass into the larva of the snail, where the germ cells continue embryonation and give rise to tadpole-like cercaria. These two asexual stages in the intermediate hose allow a single miracidium to produce as many as 250,000 infective cercaria larvae. Cercaria larvae escape into the water and swim until they encounter a fish of family Cyprinidae. They then bore under scale into the fish's muscles. Here, cercaria lose their tails and exist as metacercaria. If a mammal eats raw or undercooked infected fish, the metacarceral cyst dissolves in the intestine and young flukes apparently migrate up the bile duct and where they become adults. Now the topic has ended. Please subscribe my YouTube channel Biogenesis by Madhurima for playlist B.SC Zoology Honors M.SC CSNet Life Science and also class 9 to 12 for biology.